Okay, good morning, class. How are you today? So this morning we will start with the module 2 discussion. It is entitled Microbial Kinetics and the different types of fermentation. Okay, so before I proceed with the lecture, I would like to announce that I will be giving you a quiz for the module 1. And also, um, of course, as we do our module 2, there will be pre-test quiz, and you will answer it in the BSUEE, since we are not meeting um, uh, live or something virtually, but this is just a recorded video, so I have to require you to answer the pre-test quiz on the VSUEE. So remember, it's not recorded, but it is just a requirement in order for you to access the video lecture. So the pre-test quiz will serve as a diagnostic test or diagnostic quiz for you to, in order for me to measure your understanding or your knowledge about the topic that I have to discuss. Okay, so we will answer this the next week, so that after the lesson 3 of this module 2. Okay, so I will now share my screen, class. Mhm. Okay, so I will just run down the questions that you have to answer in the BSUEE, so that it's still part in the video lecture. Okay, so it is just true or false. You have to write this true if it is if the statement is true, or otherwise if the statement is not correct, you have to write false. Okay, number one statement: continuous culture is carried out in a closed culture system which contains an initial amount of nutrients. Is it true or false? The second statement: during the stationary phase, the microorganism appears to have no growth, but there is only minimal increase in cell density. So write true if the statement is correct. Now write false if it is not. Okay? So I guess I have to increase my volume. Okay? So the third question rather. So in batch culture, the log phase or exponential phase is when the microorganism is growing at its maximum specific growth rate. Okay, is it true or false? Number 4, which is second to the last statement, fed batch culture is similar to the batch fermentation process, but there is an extension of the batch culture by feeding periodically with medium with no removal from vessel. Is it true or false? For the last question, in continuous culture, the nutrient feed should be equal to the unused nutrient of the vessel. Okay, so that's it for the pre-test quiz. Okay, so as a review, I will just have to run down to you about the bioreactor. We have discussed the definition of bioreactor, and also the different types of bioreactor. So as a review, I will just define with you about the bioreactor. So it is a vessel or container in which the biochemical process is being carried out. So it contains microorganisms or living cells. It's not just microorganisms, but also plant cells. It could also grow animal cells or biochemically active substances derived from such microorganisms in an aerobic process or anaerobic process. Okay, so another definition, the function of which is to convert any starting material or raw material of substrate into some product. Okay, the conversion occurs through the action of the biocatalyst, which is enzymes that are produced by the microorganisms. Okay, and then these enzymes will degrade the nutrients in order that microorganisms will have a nutrient in order for the biochemical process. They have to gain energy. They need ATP in order to grow, in order to proliferate. Okay, so in order for it also to produce such a specific compound, so they undergo different biochemical processes. Okay, cells of animals and plants or subcellular structures such as chloroplasts and mitochondria. Okay, so um, the most critical condition of the requirement of the microorganism or the cells, is the substrate. It could be a simple organic chemical such as sugar or inorganic chemical such as carbon dioxide or poorly defined complex material such as meat and animal manure, or also the byproducts of the processes. For example, in the sugarcane processes, there is molasses, there is wheat bran, such as that, in order to um, make or use that for the production of such specific um, protein or such specific enzyme by the microorganisms. Okay, different bioreactor designs are needed to accommodate the great diversity of substrates, okay, products, and biocatalysts and the different requirements for the different bioconversion processes. So as we all know that we have different types of fermentation also that we can also in a submerged fermentation in which you have to the medium is in a liquid form in which the substrate are dissolved in the liquid form. There is mostly water substances. Okay, while you can also grow the microorganisms in solid state fermentation. Well, it depends upon the type of microorganism that you have to use. Of course, I am well versed with the microorganism part, so I will always have example for this about the microorganism. So you can use yeast, you can use mold for the solid state fermentation. Okay. And also, so these are the parts and function. These are the parts of the bioreactor. Okay, so I have already discussed it with you. So these are important functions, but I will just summarize with these things. These are the most important factors in which I think I have not, some of these I have not included in the previous lecture. Okay, number one is the cooling jacket. So the fermenter is really fitted externally with a cooling jacket in which through which steam for sterilization or cooling water is run. So it is necessary because um, to remove the heat generated. Okay, so it should the medium should be the temperature of the medium must be maintained in order that there will be the temperature is will not affect the growth of the microorganisms. Okay, so there should be removal of the heat generated. So this is for the successful completion of the fermentation in the fermenter. Bana IG temperature probe, so it must be maintained, and also the cooling jacket will serve as the regulation for the maintenance of the specific temperature needed for the microorganism during the fermentation. Wait, for very large fermenters, such as when you really go to pilot plants and really, if you can imagine, if you there is really production of alcohol, such beverages, but they use yeast for that, for the production of alcohol, like in San Miguel Corporation. For very large fermenters, insufficient heat transfer takes place through the jacket, and therefore there should be internal coils that should be present in the fermenter that is provided, which either steam or there is cooling water that should run. Okay, so that it's really relative to the volume of the fermenter. So it depends, because as the volume of the fermenters fermentation will take place, so there will be adjustment of different kinds of the function or the parts that should be adjusted. Okay, so there will be different designs for the different volumes of the fermenter in order to meet the requirement of the critical conditions for the fermentation. Okay, so second one is the aeration system, like the sparger and the impeller. So it is one of the most critical part of the fermenter, and in this when there is high microbial population density, so there is a tremendous, there is a high oxygen demand by the culture. So, and that is why the oxygen is being poorly soluble in water, in which there is a hard transfer is throughout the growth medium. That is why there is a needed for sparger and impeller for equal distribution of the dissolved oxygen throughout the medium. So it is necessary that there should be a good aeration system to proper for the proper aeration, in order that the microorganisms will have an access of the oxygen in the medium. So the sparger and impeller are the um, devices that are used, that are attached, that are present in the bioreactor. So the specific function of the sparger is, these are just series of holes in a metal ring. So I will go back to the previous slide. This is the sparger. So this is just a metal ring, and then there is bubbles that will introduce to the medium. There are bubbles that will be introduced to the medium. So that's for the dissolved oxygen. Okay, so there is filtered sterilized air that are introduced into the fermenter under high pressure. So the air enters the fermenter as a series of tiny bubbles from which the oxygen passes by diffusion into the liquid culture medium. So that's the function of the sparger. So another device used for the aeration system is the impeller or the agitator. So this is for the steering of the fermenter, for the uniform access of the nutrients to the microbial cells. Okay, it accomplishes two things. One is for the mixing of the gas bubble in which the sparger uh, uh, which the sparger are which the sparger will introduce. So the bubbles came from the sparger. Okay, so it mixes the gas bubbles through the liquid culture medium, and it also mixes the microbial cells through the liquid culture, okay, medium. In this way, the steering ensures uniform access of microbial cells to the nutrients. Okay, so the size and position of the impeller in the fermenter depends upon the size of the fermenter. So it is proportional. The size of the impeller depends on the size of the volume of the fermenter. Of course, if you are using a large fermenter, you should have to use also, there is also, of course, there is also a design for that, because I think it's really a package already that when there is a size, there is a particular size of the fermenter. So you have also this particular size that is fit for the bioreactor. Okay, in tall fermenters, there is more than one impeller needed for adequate aeration and agitation for it to be obtained. Okay, the ideal size of the diameter of the impeller should be 1/3 of the fermenter's diameter. So fitted above the base of the fermenter in order to have a kanang mustir gid hangtod gid sa babaw ang imong ferment ang in medium, because if you are using tall fermenters, there will be a tendency that the liquid above will not stir anymore, because naalag niya if your uh, impeller is not really fitted for that particular bioreactor. So could be no uniform access of the nutrients to the microbial cells. Okay. Um, Okay, so next, the baffles. The baffles are normally incorporated into fermenters of all sizes to prevent a vortex and to improve aeration efficiency. Okay, so they are metal strips, roughly 1/10 of the fermenter's diameter, and attached radially to the walls. Okay, so let's go back to the, so this is the, I will take the baffle. Where's that? So I think it's not here. Um, yeah, okay, it's not here. So later I will just show you the baffle. Okay, they are metal strips, roughly 1/10 of the fermenter's diameter, and they are attached to the walls of the fermenter. This is also for the improvement of the aeration efficiency. So the fourth one are is the controlling devices for the environmental factors such as pH, temperature, oxygen concentration, the nutrients, the product concentration. So um, as I have already discussed with you, that there are pH probes, temperature probe, there are also oxygen probes levels in order to maintain the levels of these conditions in the fermenter. Okay? They are monitored also via computer. Of course, it is already automated and computerized. So it is necessary not only to measure growth and product formation, but also to control the process. So that is why the bioreactor are designed for the controlling of the process of the environmental condition for the higher yield of the microorganism of the particular product that is needed. Okay, so by altering environmental parameters as the process proceeds. So for this purpose, there are various devices that are used in the fermenter. Okay, so there's, of course, the bioreactor is already computerized. So when you purchase the bioreactor, so dala na ang mga computer mga hard mga CPU, mga desktop, so appeal na siya because they are really useful for the maintenance and also um, you have to input the specific requirements of the conditions of your fermentation. Computer technology has produced a remarkable impact in fermentation in recent years. So the computers are used to model fermentation process in the industrial fermenters in order for also to prevent mga human errors. So that is why computer is really a good um, innovation. Of course, as we all know that now, so computer is really helpful for us to make our life easier, and in the same way also for the bioreactor processes. Okay, so it is really used for the process monitoring, data acquisition, data storage, and also error detection. Okay, so there are many more uses of the computer in the bioreactor. So that's it for the review of the bioreactor. So this is the module 2, the microbial growth kinetics of culture fermentation. So there are three lessons that we have to tackle. Um, Number one is the batch culture process. Second is the fed batch culture process. And the third or the last lesson is the continuous culture. So for this morning, I will just focus on the lesson one, which is the batch culture process. So at the end of this lecture, you have to define the batch culture fermentation, understand the microbial growth kinetics, and the last one is to familiarize the advantage and disadvantages of the batch culture fermentation. So let us define the batch culture fermentation. What is batch culture? Now, it is a type of culture operation that is operated also in the bioreactor, so which contains an initial limited amount of nutrient. So as the inoculated culture or inoculum cells grow, it will undergo several phases in the growth curve. So later we will discuss it. The microorganisms will follow a pattern, which is the growth curve. Okay, so the bioreactor is first charged with medium. Of course, you have already seen last week how to assemble, set up the bioreactor. You have to sterilize the medium first, and also you have to revive the cells in order for that to when it is already inoculated to the bioreactor, um, this will be active, and this will really have a good performance for the fermentation of the particular products. The cells achieve the required density or optimum product of concentration. Okay, so the bioreactor is first charged with medium inoculated with cells, and the cells achieve the required density or the optimum product concentrations. Okay, the bioreactor contents are discharged, and the bioreactor is prepared for a fresh charge of medium. Okay, since it is named as a batch culture, so first it is inoculated with medium and then let it ferment, and then after that you have to decontaminate. You have to you have to harvest also the cells, the products, and then also uh, discard the medium that are not really useful, and also you have to um, do the contamination process in order for that bioreactor will be again used for another batch of fermentation. So that's uh, um, the whole concept of the batch culture process. So there are stages of batch fermentation. One, you have to in a shake flask, of course. When you do experiment, you have to start in a shake flask first, and also you have to condition. You have to set the uh, performance or the condition of your microorganism, in which you have to start it in a small scale volume of your fermentation medium by the microorganisms, and then gradually you have to level up the volume of your fermentation medium, and also of course you have to inoculate the microorganisms proportional to the volume of your fermentation medium. You have to inoculate, you have to prepare necessary volume 10, which is 10% of the total volume of your fermentation medium. Of course, yeah, you have to inoculate that. Of course, your microorganisms should be grown in a liquid medium, not really in the solid surface na, but in a liquid medium, so in a broth. Okay, and then after shake flask, you have to inoculate your microorganisms in a seed fermenter, which is an upscale from the shake flask. It could be 1 liter and 2 liters, could be 500 ml, ganon. When you really do in a pilot scale na production fermenter, maybe it's 5 liters to 10 liters until sa mga pilot plant na mga um, okay, that's it for the stages of the batch fermentation. So the microorganisms follow a specific growth phase with the batch fermentation. Okay, so these are the growth phases that the microorganisms has to follow, or they are really doing that. Okay, in the fermentation process. Okay, so first the microorganisms will really have or in the mode of the lag phase, in which later I will just one by one discuss about it, and then you will under the microorganisms will undergo exponential phase, in which the microorganisms will actively grow, and then there will be fast consumption of the nutrients inside the fermentation medium, and also there is an active growing, exponential growing of the microbial cells. Okay, so and then after that, there will be stationary phase, in which the the growth rate of the microorganisms is equals to the death rate of the microorganism. Of course, when there is the nutrients are depleting in this stationary phase. And then after the stationary phase, there will be now the death phase. The microorganisms are slowly dying, and the but there is now no or there is minimal increase of the microbial cells because of the depletion of the nutrients, and it's all really because of the nutrients that are present in the bioreactor in this batch fermentation. Go. Okay, so our operation is thus characterized by the 3 periods of time. First is the filling period, in which you have to inoculate the microorganisms to um, the fermentation medium. Then you have to it will then undergo cell growth. Those are the phases of the microorganisms that undergo the fermentation in the batch process. And there is also cell production period, and then after that there is now the death phase period. So you will now harvest the liquid, the product, and of course it will undergo downstream processing, purification, decantation, and isolation, recovery, ganon. So we'll not dwell on that, but we'll focus on that in second sem. So there's now the final emptying period. So that's now for the downstream processing. So these are just the raw materials that are commonly used for the production of many compounds. Okay, for example, you have the carbon source is the glucose. So this is a defined composition. So actually there is also a price range, and the source of this are starch. Okay, so you can also use starch, corn syrup, high fructose corn syrup, molasses. But the source of it is are sugar beet and sugar canes. So cottonseed flour, corn stip liquor. So all of these, there are many raw materials that can be used for the production of the particular compounds by the microorganisms. So, okay, so let us now go to the phases of the batch culture process. Okay, first is the lag phase. Okay, that's the beginning of the phase of the microorganisms, in which when the microorganisms are inoculated to the fermentation medium, it will experience a lag phase. So what happens to the lag phase? What happens to the microorganisms in the lag phase? So after the inoculation of the microorganism into the fresh medium, so it will undergo a period that may be suspected that there is no growth taking place. So there is only a minimal increase of cell density. So what's the reason for this? So there is still a period of adapting or adjustment of the microbial cells to the new environment. So I guess it is just applicable for us when when we go to another place. We're adjusting so many things. So the microbial cells are still in an adjustment period. So they are not the cells are not dying there, but there is just a minimal increase. So it's negligible. So there is still no yield of the product that are taking place in that, and then also the microorganisms are still um, in their lagging phase. They're just um, they're just still there, and of course there is minimal increase of cell density, but it could be there is not, it's negligible. Okay, so in a commercial scale, the lag phase duration should be shortened as much as possible. Okay, it can be resolved in using a suitable inoculum. So in a pilot scale, in a large volume scale, no, so in order to save time for the fermentation process, because it will take place for a particular time. So when you really use it for a large scale volume now, so it is not ideal for commercial scale. So because it would take a lot of time, of course. And okay, so if we put it in an equation, in a monod equation, so the dx or dt, or the cell concentration over the time, or the substrate concentration over time, are essentially zero. So this is the yield coefficient in which there is now the production of the product, the substrate concentration over time. So there's no yield, so it is still zero, okay, in that particular phase of the microbial growth. Okay, so as you can see, if you want to put it in an equation, so y is the yield coefficient, and the x, the dx, is the oh, mass of new cells, and then also the S is the mass of substrate consumed. So there is zero yield coefficient at that time, because there is just a minimal increase of the cell density, so which is could be negligible. Okay, so for the specific growth rate, there is still no specific growth rate at the time. So it is represented by a u, or so the equation of the growth specific growth rate is the change of the cells, the mass of cells produced over the original mass of cells times the time. And also there will be another 1 over time multiplied by 1 over the time, okay, a specific point of time. Okay, so x equals the original mass of cells, and the tt is the time, change of time. Okay, that's the lag phase. Okay, let's now proceed to the log, or the exponential phase. So after the adjustment of the microorganisms to its new environment, they are now ready to grow at a maximum rate. So there is now a specific growth rate. So the growth rate of the cells gradually increases. That is why it is named as log or the exponential phase, because the growth of the microorganisms are really active and it's increasing exponentially, diba? So 2 squared, so let me say 2 squared is equals to 4. So 4 squared, so ganun, 16. Ganun. So exponential as the time increases, okay, as the time goes by. So the nutrients here are in excess and the organism is growing and its maximum specific growth rate, which is represented by a U max for this optimal condition. So in this case, the or microorganisms are already have already adjusted, and also the nutrients are being consumed also uh, in this time, because there is an increase of the uh, growth of the uh, microorganisms. They're actively dividing. So the process of the exponential growth of the microorganism is replicated through the binary fission. Okay, so as it is a review of your microbiology. So the growth of the microorganisms will continue indefinitely in the culture. Okay. So, however, when the growth of the microorganisms and the excretion of the microbial product depends on the consumption of nutrients, it dramatically affects the organism's growth, because there's only limited amount of nutrients in a batch fermentation. Of course, it really depends. There is now the limiting factor, on which the nutrients is the limiting factor to the growth of the microorganisms. When there is now no nutrients that are available in the fermentation medium, of course, the growth rate of the microorganisms will gradually decrease, because of course there is now excess microorganisms that can be consumed for the its metabolic process, no. So one siya, there's the nutrients is the limiting factor for the growth rate of the microorganisms. Okay, so after a particular time, the growth rate will gradually decrease and then its growth stops. So the termination of growth is due to the exhaustion of some essential nutrients in the medium. And there is also toxin accumulation, diba? As the microorganisms will grow, there is really production of primary metabolites that are really useful for its growth, diba? And after some times of its growing, there's now depletion of the nutrients. So there's already now the production of the secondary metabolites, in which the secondary metabolites could be toxins. These are not useful, and usually these are just defense mechanism of the microorganisms in order to thrive, in order to the other microorganisms, and there are in their natural niche. So sa ilang environment, natural environment ni siya. So it goes the same with the fermentation process, no, na there's really also the production of the secondary metabolites. It could be antibiotics, it could be mga phenols, just like that. These are can be toxin limiting. It can be toxins that are not useful for its growth, and it also and other compounds will affect its the cell's growth and could be factor for the termination of the growth of the microorganisms. Okay. So if you put it into the equation, so the cells are dividing at a constant rate, resulting in an exponential increase in the number of cells present. Okay, so there is now the mass of cells over the time equals, this is the specific growth rate, in which the KD is the cell that KD is represented by the cell death rate, and then the X is the cell concentration. So the specific growth rate minus the cell death rate times the cell concentration. This is the yield coefficient, dx over dt. Okay, so the term U minus KD can be referred to as the U net, in which the death rate is sometimes neglected because uh, the cell growth rate is really the highest in this particular phase of the microorganisms. It is considerably smaller than the cell growth rate. Okay, so there are other models used to determine cell growth rate, and it depends upon the inhibition. What kind of inhibition? There are three specific inhibition: the substrate inhibition, the product inhibition, the toxic compounds inhibition. Okay, the type of inhibition causes mathematical changes in the previously presented monad equation for the batch kinetics. So let us define this, have a discussion, a quick definition of some of the substrate inhibition. So in the batch fermentation, so in the initial growth phases, so the substrate concentrations are really high. So that is why in the batch fermentation process, it is really ideal that there is just a limit, there is uh, just the right amount. The nutrients are just limited to batch, because it could be a problem. There will be inhibition that the microorganisms will not really use, they will not really consume it right away. So it will then have an inhibition for the, for example, for the enzymes, diba? As we kanang sa review ni sa enzyme kinetics, when there is so much substrates, there will be enzyme inhibition. So it could be like that, that um, there will be no growth. There is an effect of the high concentration of substrate in the cell growth rate. So if this is a major problem, so the continuous or fed batch fermentation methods should be considered. Later on, I will discuss on the fed batch fermentation and the continuous fermentation. Okay, so for the product inhibition, so this can occur also in the induction of the recombinant gene in a recombinant microorganism. Diba? So as a review in your InBt121, diba, siya ang fundamentals of your recombinant DNA technology. So for example, you are using the E. coli, recombinant E. coli, and you have that foreign gene that codes for the insulin. Okay, so the insulin gene is being cloned in the plasmid, or introduced in the plasmid in the E. coli, no? So the plasmid, this vector is transformed into the E. coli, and then this recombinant E. coli will then express this insulin. Okay, it will then be cultivated and inoculated in a particular medium, so that the insulin will then be expressed out of the medium. Okay, so when there is now product inhibition, so um, the product itself will be an inhibitor for the, that it will terminate the growth of the microorganisms. So the products will be the limiting factor of the microorganisms in order for it to stop the growth of itself, diba? So mana siya ang product inhibition. So that's those are these are this is the second factor for um, the growth rate, that affects the growth rate of the microorganisms. Of course, since the toxic compound inhibition, when there is accumulation of toxic compounds, there is a high chance and tendency of the microorganisms that will die because of its own toxin compounds that are present. And there is really accumulation. There is now a high concentration of the toxins, and that is why there is now the death phase that will occur through time. Okay, so this is now the third phase, which is the third major phase of the microbial growth, that the growth rate has declined to zero, but it's still metabolically active, and this is now the start of the production of the secondary metabolites. So it occurs that the number of cells dividing and dying is an in equilibrium state, meaning this number of cells dividing is equal to the number of cells dying. So there is now an equilibrium state. So it can result in the depletion of one or more essential growth nutrients, and also the accumulation of the toxin by the byproduct. Okay, so that's one. These are the factors that affects the stationary phase. So because of that, there is now exhaustion of the nutrients because of the high number of the cells that are produced during the exponential phase. So the nutrients are really exhausted already, are depleted. So there's now the start of the stationary phase, in which the cells are still dividing, but then there is now there are also cells that are already dying because of um, no available nutrients that are being fed into the medium. Okay, because in the continuous and fed batch fermentation, there is feeding of the substrates during, so there will be no stationary phase that will occur in the continuous fermentation. So that's it. So it occurs when the number of cells dividing and dying is in equilibrium, and it can be the result of the following. Okay, the depletion of one or more essential growth nutrients. Second is the accumulation of toxic growth associated byproducts. And third is the stress associated with the induction of a recombinant gene. So this is for the recombinant microorganisms. So there is now the termination of the primary metabolite production. So there is no primary metabolite that are produced already, but the secondary metabolite will then take place, so I will again mention the primary metabolite that will then production of the primary metabolite will then be stopped, but the secondary metabolite will continue to produce in the stationary phase. Okay, for the last phase of the microbial growth, it is then the death phase, in which the rate of cells dying is greater than the rate of cells dividing because of the depletion of the substrate. Okay, so the decrease in growth rate and the cessation of growth due to the depletion of the substrate may be described by the relationship of between the specific growth rate and the residual growth limiting substrate. So the cells are already high concentration of cells are already dying. So this is the mathematical equation of the first order kinetics as the following, in which the change of the cell concentration over time is the minus KD, which is cell depth times the cell concentration. So there is no specific growth rate, in which negligible na ang iyang growth rate because of the high concentration of the death of the cells dying already. Okay, so as a summary, this is now the picture of this is the y-axis is the biomass concentration, the x-axis is the time in which when the microorganisms are freshly inoculated in the fermentation medium, it will then undergo the lag phase. There will be adjustment to the cell's new environment, new conditions such as temperature, pH, and even nutrients. And after that, there will be the log phase or the exponential phase, in which there is the specific growth rate is its maximum, and the cells dividing are really greater than the cells dying. So there is also fast consumption of the nutrients. The nutrients are in excess. So, and then after that, okay, after that, for the log phase, there's now the deceleration phase or the stationary phase. Okay, so this is now that the rate cells of dividing is equals to the cell's rate of dying. Okay, after that, so this is also uh, there is now no primary metabolite that are produced, but then the secondary metabolites are continue to be producing. Okay, so the secondary metabolites are produced by the microorganisms. After the stationary phase, it will then the line will then downward, in which that there is now the cells are already dying, so through time. So this is now the death phase. So as you can see that the exponential phase is uh, the highest phase in which the cells are uh, in highest concentration. So that's it. That's for the state. And then after log phase, there will be now it will now undergo to the stationary phase and then the death phase. Okay, so now later I will discuss that. So there is also the specific ways for the determination of the microbial growth. It could be via viable cell count, and also you can also do the optical density using UV-Vis spectrophotometer. Okay, so for the viable cell count, it really represents the number of cells that are actually viable, okay, that are really growing, and it could be determined by a pore plating. You have to do microbiological technique. You have to do serial deletion. Okay, you have to get a sample from the flask that the microorganisms are already there. Okay, you have to get a sample about 10 ml, and you have to do serial dilution. And after that, you have to plate the uh, the these samples from the particular dilution. Okay, for example, 10 to the second, 10 to the fourth, 10 to the sixth. Okay, after that, you have to count the colonies that are present, that are grown sometimes after the incubation period. If it's microorganism or if it's bacteria, you have to wait for 24 to 48 hours in order to count the colonies that are already growing in there in your plates. Okay, while the optical density, so it is the simplest and the fastest one to obtain the concentration, the cell concentration. So initially higher curve representing the number of cells that are both viable and non-viable. So it is determined through an optical measurement using a spectrophotometer, okay, using turbidity. Okay, so optical density, it is the quick and easy way to develop a growth curve, and one takes a sample of the fermentation broth and then measures the absorbance at a particular wavelength. Sorry. Okay, so for the E. coli cells in typically LB medium, so the wavelength is you have to set the wavelength in the spectral photometer It's in 600 nanometer. So it is the same with the Bacillus apilis. It is also the same with the yeast. So I have experience doing that, and during my thesis, during my experiments in my MS. So yeah, that's a common wavelength for the bacteria. So as you can see that there will be different turbidities you you can observe during the growth of the microorganisms in a particular time. Okay, the measured value can be compared to previous measurements made in conjunction with the cell plating or cell counting. The negative side of this um, spectrometer photometer, the optical density determination, is that you will not the absorption of the wavelength is for the non-viable and the viable cells. So, but for the viable cell count, you are really assured that it is really viable. The counts are for the viable cells. So the values taken are not representative of only viable cells. So it is also recommended that uh, you have to conduct both to ensure um, the viability of your microorganisms. So, and that's it for the lesson one. Okay, so this is the learning task. So using a block flow diagram, present the general fermentation process in a batch culture with short explanation in your own words of each block. Cite references used for the explanation. So you can really search the internet for the batch culture with short explanation. Okay. So that's it, class. So for the next meeting next week, I will discuss with you the lesson two, which is the fed batch culture. So I will now stop share. Okay. So thank you so much, class, for watching this video, and I will post the quiz later at 12 noon sa VSUE. So thank you so much, and I hope and I pray that you are safe at your homes and you are well and good. Thank you so much. God bless. Bye-bye.